Hi, everyone. I am Arissa. I am a member of the Women in STEM Club. And today I have the honor of introducing Cynthia Chen, who is a freshman at Harvard and graduated Harker last year. She enjoys exploring research in the fields of deep learning, computational biology, and statistics, and is particularly interested in developing methods to explain AI models better. Today, she will be speaking to us about decoding neural networks. And before we get started, if anybody has any questions throughout the duration of the talk, please just direct them to the private chat and we'll get to them in the last 5 minutes or so. All right, Cynthia, take it away.
Yeah, thanks, Arissa, for the intro. I'll go ahead and share my screen right now. Okay, cool. Okay, cool. Hi, everyone. My name is Cynthia. I'm a current freshman at Harvard studying CS and statistics. And today I'll be presenting my project, which is titled Decoding Neural Networks, Discovery of Anti-Tumor B-cell receptor motifs using a novel sequence-based computational framework. So one of the biggest questions within the field of cancer research is how can we genetically differentiate between various cancer types, such as breast cancer, prostate cancer, and lung cancer, for example. And to focus on this question, we address the B-cell receptor, which is a part of the human adaptive immune system. And so the B-cell receptor plays a really crucial role in our immune system because it has these receptors that are able to identify specific antigens correlated with certain tumor cells and cancer types. And because of this, it's able to signal the release of antibodies to attack these tumor cells. And so in order to better understand what sorts of antigens are correlated with certain B-cell receptors, it is really crucial to identify these tumor-specific sequence patterns on the B-cell receptor, which are termed motifs. So previously, my mentor developed the DPCR model, which is a 6-layer deep neural network, as you can see here. And so the DPCR model was trained on more than 30 million B-cell receptor sequences from 5,000 patients. And after model training, it was able to achieve a really high accuracy in predicting what cancer type a certain B-cell receptor sequence belonged to. And so this high accuracy prompted us to ask the question of what exactly did the model learn from the training data that allowed it to make such accurate predictions? And how can we extract this information? So currently, although artificial intelligence has made really huge strides within the last few decades, it remains a black box, meaning that there are no set and rigorous methods to extract the information that artificial intelligence models learn from training data. And because of this, they're not very explainable, and we can't exactly pinpoint why they're making the decisions that they're making. And so this was one of the goals in our project, was to create a framework to decode the DPCR model and see what it learned from the data that allowed it to be so accurate in making these classifications. So far I've presented two problems. The first was how can we genetically differentiate between various cancer types? And the second is how can we develop methods to better understand artificial intelligence and deep learning models? And so my research lies at the intersection of these two problems. Namely, by decoding the DPCR model, we're able to identify these tumor-specific patterns that will allow us to create more targeted and specific cancer medicines. And so the goal of my research in the first year was to develop such a pipeline for decoding the DPCR model and identifying what sort of motifs it learned from the training data. And in the second year of my research, I aimed to generalize this pipeline so that it was applicable towards all all deep learning models trained on sequence datasets. So in order to accomplish these objectives, we developed a novel computational framework, as you can see here. And so pretty much how the framework works is it generally, it first generates a bunch of random sequences and then inputs them through the DPCR model, and then looks at these tumor-specific output scores for each of the cancer types. Based on these scores, it ranks these sequences for each of the cancer types to identify the highest scoring sequences for each of the cancer types. Then, based on these sequences, it performs clustering and visualizations in order to identify the most prominent patterns. And so once these patterns and motifs are identified, we then performed validation using correlation analyses and binding site analyses in order to make sure that they are the correct motifs. And then furthermore, we generalized our pipeline towards all deep learning models trained on protein sequence data and applied it to a second deep learning model called the DeepDagron model. So when we applied our framework to deep BCR, we were able to identify 65 anti-tumor B-cell receptor motifs, as shown here. And so the background of each cell basically represents how prominently enriched it was among our analyses. And so when we took a further look at this table, we were able to identify some pretty interesting patterns. So first, when you look at the BRCA, which is breast cancer, and PRAD, which is prostate cancer, if you look at those two rows, you can see that they share this highly enriched DPSDC motif. And so this is pretty interesting because a previous study actually shows that breast cancer and prostate cancer are linked within families. And so the fact that they're both really highly represented within our analyses makes a lot of sense. Furthermore, if you look at the LUAD and LUSC rows, which represent two different types of lung cancer, you can see that they share the top 5 highly enriched motifs, which also makes sense because we would expect two types of lung cancer to have similar protein sequence patterns on the B-cell receptor. So these sort of qualitative analyses validated our motifs, but we also wanted to perform some quantitative validation analyses as well. And so to do this, we looked at 2 different DPCR models trained on different model parameters, trained them on the data set, and then performed our framework pipeline analyses on both of them. We then analyzed their tumor-specific correlation score. And so as you can see in this correlation heat map over here, the correlation along the diagonal is really strong, which shows that even when 2 different models were used, the results for the same cancer type were very similar, which validates the tumor specificity of our motifs. And so furthermore, we were able to conduct training iteration analyses where we looked at models at different steps of the training process and then saw what sorts of motifs they generated and their overall correlation score. And so as you can see in this 3D diagram over here, the highest correlation was achieved at the point where model 1 and model 2 had the highest iterations, which validates the robustness of our model training. And so in the second year of our research, we generalized this pipeline and then applied it to a second deep neural network called the DeepDagron model. And through this analysis, we were able to identify some specific protein mutations that lead to the most protein degradation. And so the whole point of this experiment was to basically demonstrate the generalizability of our framework and show that it's not just specific to the deep BCR model. So in the end, we were able to draw 3 major conclusions. First, we both identified and validated 65 anti-tumor B-cell receptor motifs that are potentially implicated within this B-cell receptor and antigen binding interaction. And we further validated these motifs both qualitatively and quantitatively through correlation analyses. Furthermore, we conducted a pan-cancer, multi-cancer analysis, in which we were able to determine motifs for 13 different cancer types. And so the best part about this is that our methods are not just specific to a single disease or a single cancer type, but can actually be generalized towards other diseases as well. And last, we were able to develop a generalizable computational framework for decoding sequence-based computational deep neural networks. And we were able to demonstrate the generalizability and applicability of our pipeline through an application to the DeepDagron model. And so before I end, I'd like to briefly go over the significance and impact of my work. On the biology side, our research can lead to a better understanding of how the B-cell receptor interacts with the antigen, which can lead to an overall better understanding of our human adaptive immune system. Furthermore, knowing these motifs can help doctors better predict how certain drugs will react within patients. And most importantly, knowledge of these motifs can help us synthesize more targeted specific antibody drugs, which can help precisely target a certain cancer type within patients. Furthermore, on the AI and deep learning side, our research introduces this general computational framework to identify these specific patterns learned by deep neural networks. And we demonstrated that it's applicable towards other models as well. And so this can serve as an important first step towards more reliable and explainable AI models that are trained on biological data sets. And so there's a lot of further pathways for this research, such as potentially analyzing 3D structures of these proteins and maybe conducting wet lab analyses to further validate the motifs that we discovered. So this research project would definitely not be possible without the help and support of several people, including my mentors, Dr. Sherlock Hu, Dr. Colin Tokim, and Professor Shirley Liu. I'd also like to thank Mr. Chris Benner and Miss Anita Chetty of the Harker School, as well as my family for their never-ending support. Thank you so much, and I'm open to answer any questions that you guys might have.
All right, so I haven't received anything in the chat. So if you do have any questions, go ahead and just unmute, or again, you can use that chat function.
Wait, I kind of missed the beginning of your talk. So I joined when you said you needed to use a qualitative approach too. Could you explain why you needed to do that?
Yeah, so I think you might be referring to the validation analyses. Sorry. Let's just go back to this slide. So from this table, we were able to perform some sort of qualitative analyses where we just looked at the table and identified some sort of patterns or similarities among these different cancer types. And we were able to identify similarities between the breast cancer rows and the prostate cancer rows. But this is just like a qualitative perspective where we just looked at it and didn't really calculate anything. So we wanted to perform some more rigorous quantitative validation analyses as well. So on this slide, what I'm showing is just the correlation analyses between various tumor types. And we can see that there is a very strong correlation along the diagonal, which shows that the motifs that we discovered are quite tumor specific and not just random.
Okay, that makes sense. Thank you.
Yeah, I have a question. What led you to get interested in this topic and kind of combine these two things? How did that idea come about?
Yeah, that's a great question. And I think I kind of got really interested in this topic of AI explainability and figuring out ways to better understand deep neural networks because before conducting this project, I actually conducted a research project where I built a machine learning model to analyze another biological data set, and it also had pretty high accuracy as well. And so after that, I started reading more papers within the field of AI, and there was a lot of research going on about creating models to analyze data and having really strong performance. But I noticed that there wasn't too much research done to look back at these models and almost reverse engineer them and see what they learned. And so this led me to, I guess, approach labs with this question of how can we better understand deep learning models. And this happened to be a really great fit because my mentor had just built a deep learning model. And so I was able to add on to his project by looking and examining the model a little bit more.
All right, cool. And I guess kind of an overarching question is, with these kind of more advanced topics, how would you what is your advice to someone who is just beginning and how they can get started?
Yeah, for sure. I think that's a really good question. I think within the field of if you're aiming to do some sort of computational project where either you're analyzing data or building a model or doing some sort of quantitative analysis, I think a really great way to start is to learn maybe Python or some sort of coding language. I think that having that sort of skill of CS and just being able to program something is really crucial when it comes to literally anything within computational research. So I would suggest learning Python because I think that's a really approachable language and commonly used within CS research. Beyond that, I know that research papers may be kind of daunting sometimes. So I think that Harker offers a bunch of really great resources, such as maybe the research methods class or advanced research class that have helped me a lot in improving my own research skills and just finding other students who are interested in research.
All right, cool. Thank you so much. Well, thank you everyone for attending. Thank you so much, Cynthia. And you guys can go ahead to the symposium website and look at the other phenomenal talks and activities that they have planned for us. All right. Thank you, everyone.